Single point mutations. These mutations are involved in changing a single base in a DNA strand. A point mutation impacts the primary structure of a protein. Oftentimes, these overlooked changes can cause detrimental effects on the biological processes of the human body. Point mutations can have 4 different effects on proteins: missense, nonsense, silent, and read-through. We will go over each one in detail momentarily. Before we get to that, however, let's go over the types of point mutations: transition and transversion. In transition mutations, a purine is changed to another purine, or a pyrimidine is changed to another pyrimidine. A way to remember the difference between purine and pyrimidine is cut pi. The bases cytosine, uracil, and thymine are pyrimidines. Adenine and guanine are not. Transversion mutations are where one type of nucleic base is substituted for the other. A purine is substituted for a pyrimidine, or a pyrimidine for a purine. This information will be important for the next section of the video: point mutation effects. In a missense point mutation, the single base change can now cause a new amino acid to be coded for example in this DNA strand. We see that the strand has changed the codon from AAC to AGC. The mRNA's codon originally coded for asparagine. Now, because of the transition point mutation, where the A was switched for a G, it will now code for serine. Nonsense will cause a premature stop in protein synthesis. Let's look at the codon for cysteine. The original codon was UGC. We have now changed the C for an adenine base. This codes for one of the 3 stop codons, UGA. The change from a C to an A constitutes a transversion point mutation. C is a pyrimidine and has now been converted to a purine. A silent mutation refers to the idea that a change in base will still code for the same amino acid. Here, GCU was converted to GCA. GCU and GCA both code for the amino acid alanine. The change to the A categorizes this as a transversion mutation. Lastly, in a read-through mutation, the stop codon is changed to a new amino acid, making a longer protein. Here, UGA, which is one of the 3 stop codons, has been changed to UGG. This new codon codes for the amino acid tryptophan. When this mRNA strand is translated, it will not stop and will actually continue to translate the amino acid structure. The change from an A to a G makes this a transition mutation. I hope this video has been helpful in showing the different types of point mutations and their effects. Sometimes, as seen in silent mutation, the protein structure will not change because the same amino acid has been coded for. Other times, a base change can cause a completely new protein to be synthesized, affecting its primary structure and potentially its overall function.